I want to welcome you to the second day of the NIH's Behavioral and Social Science Research Festival. I'm Christine Hunter. I'm the Deputy Director for the Office of Behavioral and Social Science Research. This year's festival honors the 25th anniversary of OBSSR. I hope you enjoyed the excellent presentations yesterday as much as I did, and are looking forward to 4 more outstanding speakers today. I want to remind you that as people are giving their presentations, you can enter your questions and comments during the presentation. So when we move to the question and answer section, we've got some queued up and are ready to go. And so with that, I'm going to introduce our first speaker. So I'd like to introduce Dr. Rick Kittles. He's Professor, Associate Director of Health Equities in the Comprehensive Cancer Center and Director of the Division of Health Equities at the City of Hope. And his talk title is Race, Health, and the Politics of Skin Color. So with that, thank you, Dr. Kittles. I'll turn it over to you.
Thank you, Christine. And I want to thank the organizers and for this honor to give a talk to this group. And today my talk deals with race, health, and the politics of skin color. And what this talk is mainly about is some long-time work that we've been involved in looking at issues of race and genetics and ancestry, and then also how in the US in particular, skin color mediates how one views themselves and how others view them, and how that impacts on certain sort of behaviors that could have social impact, but also biological outcomes. So next slide. Sorry, I was getting ready to turn it myself. So race is a crude proxy historically in biomedical research. If you look through the literature, race has been used as a proxy for some shared sort of biology and some shared environment. The problem though is when we think about complex diseases and the risk factors and the contributors, it's very hard to tease apart those two when you use race as a crude proxy. You can go through the literature, you'll see that race is a predictor of health, of a slew of different health outcomes. But it doesn't say much about what are those causal sort of factors, whether it's biology, some biomarker or some genotypes, or if it's due to some lifestyle or dietary or exposure in the environment. So what we're moving away from now is using race as this crude proxy and actually talking about ancestry, genetic ancestry, as a variable in this model. And when we talk about race, we're talking about the social component of race, social demographic groups, very useful for measuring, doing surveillance and measuring outcomes, but not necessarily that useful as a predictor of risk. Next slide, please. So as we move away from race and talk about genetic ancestry, one of the things that's very clear, and we've started doing this back in early 2000, looking at genetic ancestry in African Americans across the United States. One of the things we find is that communities of color are very biologically diverse. Next slide. And that's a lot of it has to do in the African American population due to the high genetic diversity of the antiquity of the African gene pool. So the bulk of it, of African Americans get their gene pool from Western Central Africa. And then also, to some extent, gene flow with non-Africans. And that pattern of variation differs geographically. Next slide. This slide just shows where the enslaved Africans came during the transatlantic slave trade. So these over 10 million enslaved Western Central Africans were enslaved and brought to the so-called New World and just completely disconnected from their cultures and families. And so for the most part, northern Senegal to southern Angola and East Africa, Mozambique and Madagascar. Next slide. So we can use genetic ancestry to uncover the genetic structure at the individual and also the population level. I'm not going to get very deep into the genetics, but I do want to give a good overview in terms of how we do this and why we do this. Next slide. So this is just an admixing model where we are looking at 2 putative ancestral populations that contribute genes to if you look in the middle there, an admixed population, And so at the first generation, they would have 50% of genes from one group, 50 from the other. And then over time, we get that shuffling that occurs due to recombination over several generations. We get this, what we call a mosaic of these chromosomes, some coming from one ancestral population, others from another. So here in this model, we're assuming that one of the populations is West African, the other one is of European ancestry. And so in defining sort of the ancestry of African Americans, this is a very useful model. Next. We use markers called ancestry informative markers. Lately, we've been, because of the very large number of genetic variants in these databases, we don't necessarily have to use ancestry informative markers. We can just use a large number of markers to get a really good estimate of ancestry at the global level for African Americans and Hispanic populations. Next. This is just a PCA plot showing the distribution of genetic ancestry in 4 groups. The black dots are Nigerians from Yoruba Nigerians. The red dots represent African Americans, blue are European Americans, and the green are European populations. And so you'll notice this spread of ancestry, and there's no clear sort of dividing line in terms of who's black or who's white. If you look even in the area where the blue dots are, there are some African Americans, the red dots, who genetically have more European ancestry than some Europeans, but identify themselves as African American. And this is important to understand because, you know, we can't assume that race, self-identified race, is a predictor of genetic background. There's a reason why these African Americans and why these European Americans define themselves as white or black. And a lot of it has to do with how they were socialized, how they shaped their identity over time. Next slide. African Americans aren't the only group that are very heterogeneous. Hispanic, Latino populations also extremely heterogeneous. In fact, maybe even more because these populations have 3 ancestral groups that contribute to their gene pool. So we have the Spanish European, the Native American genetic ancestry, and then African. What's fascinating is even among the Hispanics, there's enormous heterogeneity because some have a much closer history with the transatlantic slave trade than other groups. Like for instance, Puerto Rican, Dominican, and Cubans have a larger proportion of West African ancestry in their gene pool than, let's say, Mexicans. Next slide. What's really cool is now that, you know, we have markers that go across all these chromosomes, we can actually estimate ancestry along chromosomes and then leverage that to do what we call admixture mapping. I'm not going to get much into that, but I will say that if you look at this on the left side, the left rectangle, you'll notice the African Americans, there's this mosaic of blue and red among those chromosomes. This is a segment of chromosome 5. And it depends on the family history. Some African Americans have a European ancestor in a more recent generation than others. And so we see some individuals having these larger blocks of European ancestry across their genome. Next. This actually is a chromosome plot for me. So this is an estimate of ancestry along my chromosomes. And you'll notice that, in fact, I have about 90% West African, or what we call here sub-Saharan ancestry, and about 10% European ancestry. But what's really fascinating, if you look on chromosome 5, where I pretty much have all African alleles, which is in the burgundy there, the dark burgundy color, and very little, if any, European ancestry along that chromosome. While if you look at chromosome 10, a quarter of it is of European ancestry. And so this is what I mean by the shuffling, the recombination that occurs, and it creates this rich mosaic of ancestry along these chromosomal segments. Now, why I brought the attention to chromosome 15 is there are some major predictors on chromosome 15. Two of the major predictors of skin color are on chromosome 15. And I have African alleles for each one of those throughout those genes. And so that's what gives me my rich chocolate skin tone, is because, you know, while I have 10% European ancestry, the important genes there for skin color are all of African ancestry in terms of those alleles. And so we can leverage, just like I just did with skin color, when we look at diseases that disproportionately impact one group or another, we call that admixture mapping. I'm not going to get into that, but just know that this is very useful for studying genetics of complex diseases. Next slide. And this is a buddy of mine. I always tease him, Sean Kimbrough. He's at North Carolina NCCU. And I use him as an example because just like me, he's an African American. But the average proportion of European ancestry for him is much higher based on family history. And you'll notice that along every chromosome, there's large blocks of European ancestry. Next slide. So we have this high genetic heterogeneity. We can uncover at the genetic structure, at the individual level, and also at the population level. What's even more fascinating is that the pattern of variation varies geographically and socially. Next slide. You look across the United States, different African American communities, on average, it's about 20% European ancestry in African Americans. But there are some communities, some regions where it's much lower, and other regions where it's much higher. So, for instance, if you go to South Carolina, there 3.5, and right off the coast, the Gullah Sea Islands off the coast of South Carolina, it's only about 3.5% European ancestry on average among those African Americans. While all the way on the other side, on the West Coast, in Seattle, it's about 35%, which is 10 times that. And so there's enormous heterogeneity there. And that says a lot about the family history of these individuals that we consider to be African Americans. In fact, 35% means that on average, 3 to 4 of 10 individuals that were tested had one white parent, and which is pretty high, and that's why you see that very high proportion of ancestry there, of European ancestry. And of course, if you look across the South and compare to the North, the northern states, those communities, those cities have a higher proportion of ancestry than the Southern. What I call the social norms are much more restricted in certain areas where you wouldn't see as much intermixing between African Americans and whites. You'll notice that there are 4 circles there, 4 cities that are circled: New York, Washington, D.C., Cincinnati, and Oklahoma. And there's a reason for that. We actually studied those 4 in depth. Next slide. In order to understand this issue of how race should be used in these sort of studies, and when we talk about ancestry, we're talking about the genetic component. So some believe that we should throw race out of the discussion and just use ancestry and just measure the environment. That would be good. However, race does have some meaning to it. And I'll talk about that in a second, of how skin color in the United States in particular really mediates how one is viewed within and between different groups. So that's the social component. And then we have this interaction that could occur that could impact complex disease. Next slide. This is important, all of these discussions, because as we move forward in terms of precision medicine, what we're finding is that much of the data that's collected is based on populations of European ancestry. And one thinks that we can just grab a group of African Americans or Hispanics and think that they would be representative of all of those subgroups. And in fact, they're not. Next slide. Because this is, as I said, this is particularly important as one goes deeper in this movement towards precision medicine. I believe precision medicine is going to have enormous potential. However, we have to make sure that all communities benefit from it. Next slide. And also genetics, we have to be honest, genetics alone is not going to help us by itself. And as a geneticist, I learned early on in my studies of disparities that genetics is only going to provide some level of insight. Most of the action, I tell the students all the time, most of the action is in the social, cultural, behavioral sort of realm when one talks about disparities. Next slide. So when we talk about factors contributing to health disparities, gene environment interactions, accumulated stress. There was some discussions earlier yesterday about stress and how that impacts one's immune system. So weathering, allostatic load, life course selection, cultural factors, behavioral differences, discrimination and perceived racism. This is something that I think we really need to examine more, because we are not in this race-free society that many believe. we have, we all know that there's institutionalized racism that is playing a very strong role in the health of certain African American and Hispanic communities. And a lot of this is based around socioeconomic status and institutional arrangements. Next slide. Oh, this is just a chart from a paper that I really like, because what this actually says is there's a correlation among perceived discrimination and health outcomes when one thinks about how one responds to the physician. So if you have a situation where you have African American patients and the physician may not be African American, let's say even if there was no meaning overt discrimination, let's just say there was this perception of being discriminated against, it led to poor outcomes, health outcomes for the patient. And we see it all across the board where you see these negative correlations in terms of health status and team perceptions, thinking that, or the patient believing that they and their doctor are working as a team to improve health. All negative correlations. The only thing that was positive was the number of chronic diseases when one looks at the perception of discrimination that the patient may feel. Next slide. And this is something I've been working on for a bit. This is actually the top part of this circle. It actually comes from Kamara Jones, who's I just added the bottom part to really talk about what's driving many of these health outcomes. This racial and biocultural climate on health. And Dr. Jones talked about this internalized health behaviors, institutionalized SES, access to healthcare, and then also personally mediated, how one deals with stress, how one deals with differential treatment. And a lot of this is mediated by skin color, which is why I put that on the outside. And then we have the genetic component, and how genes and genes of behavior or genes in the environment may interact and lead to poor outcomes also. Next slide. So let's talk about skin color real quick. So we talk about ancestry and race. We can't ignore skin color, because skin color has historically been employed to categorize populations into these racially classified social groups and to stratify opportunity structures and life chances via racism and systemic discrimination against individuals and groups. And what's interesting is that skin color is strongly correlated with genetic ancestry. So when we do a genetic ancestry estimate, in particular for African Americans, we find that skin color, there's a correlation there with skin color. So thus, the study of ancestry and health outcomes may strongly entail both biological and also sociological factors. And so you really have to have a really robust analysis in order to tease apart those potential confounders. Next slide. This is just a plot showing ancestry on the x-axis and skin color, what we call the M index. This is from a skin color reflectance meter on the y-axis. And you'll notice that there's this correlation there. The R-squared is about 36, and it's very strong. Some communities, the correlation is stronger. Some they're weaker, but there is this correlation that's quite significant. Next slide. So given the attention to health disparities, in particular in the mounting evidence showing genetic variants associated with some particular outcomes, identifying the influence of ancestry on these complex phenotypes may allow for the deconstruction of race, as I mentioned earlier in the talk. So, however, though, few studies examine regional differences in genetic ancestry that may correlate with these social cultural factors within each region. And so they may be specifically tied to SES. And I'll get into that in a second. Next slide. So I want to talk briefly about this 4-city study that we did back in 2004 to 2006. So over 2 years, we went into 4 different African-American communities in the United States and to study ancestry and attitudes about ancestry and identity and disease across 4 different communities. We did pre and post test surveys. We looked at SES data. We collected buccal swabs for DNA and typed ancestry markers, and then also measured skin color. And then looked to see how strong were some of these correlations with ancestry and SES, ancestry occupation, let's say, and then also skin color in some of these communities. Actually, this was published recently in PLOS One. Next slide. We started this study, interesting enough, based on a request from the Black freedmen community in Oklahoma. These are descendants of individuals that were part of the Trail of Tears, what's called the Trail of Tears. 5 Native American communities that were moved and pushed away from their homeland and then moved to reservations in Oklahoma: the Cherokee Creek, Seminole, Chickasaw, and the Choctaw. This was a horrific period for those communities. But before they were moved by the European settlers, many of them had relationships and had actually had African descendants in their tribes, either as enslaved Africans, or they were actually members and in some cases leaders of those communities. Next slide. And this woman, Sylvia Davis, actually invited me to come out and do some testing to show that they had significant Native American ancestry. This is back in '04, and at the time there wasn't a lot of markers for Native American ancestry, but we did a good job in the sense of being able to identify who had significant, who had minor. But the point was they just really wanted to show some utility with this technology. And so this is why I tell people all the time, when we talk about a lot of these direct-to-consumer technologies that people are using, the utility varies according to different groups. And some groups want to leverage it for things like, for instance, Indian or Native American ancestry in the court system. Next slide. And there's a rich history of African enslaved, of people of African descent in Oklahoma, in particular Tulsa. Many of you may remember Tulsa had this enormous race riot in 1921. Next slide. And so we looked at not only Oklahoma, but we went into Cincinnati, New York City, and DC. These are all self-identified African Americans, and saw that for the most part, the genetic background in terms of ancestry was pretty similar. And in fact, Oklahoma had the lowest percentage average Native American genetic ancestry than even Cincinnati, New York, and DC, which of course they weren't that satisfied or happy with, but that was the reality. And so it was a learning experience for everybody, not just us, but for the communities in which we provided and were studying as it relates to identity. Next slide. Actually, it was written up by a popular magazine, Wired magazine, called The Blood Feud. And so this is actually where science and society intersect in very interesting ways. And in fact, one thinks that the technology can help or has strong utility, but when in fact it doesn't, you have to create opportunities for reconciliation. How do we reconcile this so-called empirical data from DNA? And a lot of it has to do with an understanding that in many of these Native American communities, they had African Americans in those tribes, but they weren't blood related. They were part of those groups because of the social structure that emerged after the Trail of Tears, not necessarily because of bloodline. Next slide. So going back to the 4-city study. Now, one of the interesting things is that when we pulled all the data together, because we wanted to do sort of like what the average large scale study does, is they put all the African Americans together, irrespective of where they came from, right? So one of the things we saw was that at the bottom there, you see SIRE, self-identified race and ethnicity. And so those who said that they were African American, if you look at that first column, that's the M index, that's skin color, right? And then the next column is West African ancestry, European ancestry, and then the last column is Native American, percentage Native American ancestry. And you'll notice that those who said that they were African American were darker in complexion than those who said they were mixed, right? So SIRE did, it does have some meaning to it, right? And it does carry some weight with it, this self-identified report, right? And then if you look at percentage West African ancestry, we also see higher proportion of West African ancestry in those who say that they're African American versus those who say that they're mixed. And it's fascinating because saying that you're mixed is one in which a situation where how you're socialized, because you could be from the same family structure in a different part of the country and never claim that you're of mixed ancestry. You would, in many cases, many say that they're African American. And so I love the fact that we identify ourselves the way we want to. And I think that's very important. And that's why I always felt that Ancestry is not going to create any fuss about defining and certifying who's who. People are going to define themselves based on the information that they have. And a lot of it has to do with how their family structure is shaped and what they know about their family history and ancestry. So then you'll also notice that skin color differences across those three communities. Actually, we weren't able to do skin color testing in Oklahoma, but in the other three cities we were. And you'll notice that there's some significant differences there in skin color. Also, percentage Native American ancestry at the top of the chart was very different. For instance, a larger proportion of individuals who were unskilled had a higher proportion of Native American genetic ancestry. I'm going to come back to that shortly. Next slide. This is, so then we broke it up into the different cities, right? And you'll notice that in Cincinnati, Ohio, skin color was correlated with occupation. And so those that had darker skin color were more unskilled or they weren't in the professional ranks as much as they were in the unskilled rank, which says something about the local history, local experiences, and the culture in the African American community in Cincinnati. Next slide. In Harlem, what we saw fall out was this Native American component. So when we pulled the data together, remember you saw that there was this high proportion of Native American ancestry in the unskilled ranks of occupation. And you'll notice that that had to do with Harlem being the driver in that pooled analysis. And we actually went into Spanish Harlem and did this. And so while the bulk of the individuals were African American, there were some that would be considered Dominican or Puerto Rican. And I think that is where we saw the high proportion of the contribution of the Native American genetic ancestry. Next slide. You'll notice with income, for Oklahoma, income fell out as it relates to ancestry, West African ancestry, and European ancestry, and Native American ancestry. So once again, we see, for instance, with European genetic ancestry, those that had higher proportion of European ancestry making more than those who had less European genetic ancestry. And then also a similar reverse with the percentage Native American, with those having the higher proportion of Native American ancestry making less. So these are all of these cities just reiterate sort of this common theme that we shouldn't necessarily look at African Americans as this homogeneous sort of group, but actually as a macro ethnic group with different local experiences and different sort of population trajectories that impact on opportunities, life opportunities, and then also health outcomes. Next slide. This is Washington, D.C. We didn't see anything fall out for that particular city. I think if you look at it, it's a very cosmopolitan city, and there was no significant correlation with ancestry and skin color and SES. Next slide. So considering the differences that we've identified across these 4 cities, it might be useful to leverage ancestry, genetic ancestry, and local historic social context into research. Doing so may provide a more robust understanding of the relationship between our genes, the environment, and disease. Next slide. So in summary, the relationship among skin color, identity, ancestry, and disease are complex and influenced by cultural norms, history, and behavior. Next. So for recently admixed populations like African Americans and Hispanics, a self-identified race and ethnicity is not necessarily a good proxy for genetic background. However, as I said, we don't throw the baby out with when we talk about race. Race does have some potential meaning, and I think we really need to see that more. And I think a lot of it is going to be mediated by how one views skin color in American society. We need to develop a study designs that recognize the influence of SES, racism, ancestry, and their interactions to minimize the confounding effects of this social environment on genetic studies. Next slide. So I'm going to end here, and I want to thank the Freedmen community in Oklahoma for their support and initiation, actually, of Four City Study. If it wasn't for them, we actually probably wouldn't have gone into the other cities. We really need to study and compare across these four cities. Also, the National Underground Railroad and Freedom Center Museum in Cincinnati, and then in Harlem, New York, the Church of Latter-day Saints, and at Washington, D.C., the National Genome Center at Howard, and also my collaborators for that particular study. So, thank you, and with that, I'll take some questions.
Thank you so much, Dr. Rick Kittles. That was a really interesting talk, and I think it showed the biological and social impact of race and suggested that there may be differential impacts of skin color and genetic history affecting these complex genetic diseases and disorders. So we do have time for a few questions now. So one question that we received was, based on your study, how do you propose that researchers, for example, as those looking at the impact of race and ethnicity on health outcomes, should overcome the possible contradiction of self-reported ethnicity versus that which is dominant in their genetic ancestry? And what do you see as the potential risks of moving away from the connection of race and identity?
Right. So great questions. And so when we talk about self-identified race and ethnicity, it's an important variable. I think any study, you should have a demographic questionnaire where you ask these individuals. You don't have the study coordinator define who they are. You ask the individual, how do you define yourself? And in many cases, they'll say African American or Hispanic or whatever. But then ask them how they would define their parents and their grandparents, because then you really get to see sort of the mixture that could occur within that household, within the family structure, but yet also see how they identify themselves. And so that's why whenever we do our studies, we ask the individual, and then we ask them how they would define their mother and their grandparents. And then when we look at the genetic ancestry markers, it allows us to even to group even better, and to be able to justify why one individual is put into one group versus the other. I also think that race, we need to do a lot more work on race. I think we really need to study a lot more how race is, why is it so salient in American society still today? And with that, what is it carrying with it? What is the information that it's carrying that could impact one's health? And people say, oh, well, it's just a proxy. Well, if it's just a proxy, and that proxy is defined by skin color, and we know skin color is how one stratifies these racial groups, then there's some significant impact in that proxy. And so I think we really need to do more on that, more research in that area.
Great, thank you for that response. Along the similar lines, and we're talking about translating this to more of a lay audience, how might we help people understand both the difference in value of both ancestry and self-identified race? And how does this change how someone defines themselves once they find out their genetic background? Does this differ by race?
Right. So I don't think most people change anything about how they feel when they find out their genetic background and genetic ancestry. I think by the time they've done that this sort of testing, or they've found out their ancestry, they've already formed their identity. They've already pretty much created this identity for themselves. And in some cases, it might jar them. It might create some discomfort or room for reconciliation, but it doesn't necessarily, they're not, I don't think they're going to have many, or major changes in their life. And this is based on data that us and others have accumulated. Self-report and is important because it allows you to sort of group individuals together. You could say, okay, we have African Americans in this group. Once you look at ancestry in that population, you may find that some of them may not necessarily reflect that genetic background. You might want to leverage ancestry more in your statistical analysis, or you might want to throw out certain people because they are sort of like anomalies. So there's much you could do to leverage both of them at the same time. Ancestry is important for I didn't put these slides in my talk, maybe I should have, but as we move further along in precision medicine, ancestry is going to play an important role in terms of taking us one step further towards individualized medicine or personalized medicine. Many drugs that are emerging now that have these labels that are saying, you know, this people of African descent or people of Asian descent may not respond that well to this particular drug. But then come to find out that, you know, even within those groups, there are groups of African Americans that respond to this drug. We did a big study in Chicago where we looked at naltrexone, which is a drug for smoking cessation. It used to be for alcohol abuse, but now they were using it at the time for smoking cessation. And the literature said that whites respond better than blacks. And so there was a lot of discussion around, let's not give it to African Americans because they're not going to respond to it. But then we did this study where we actually stratified African Americans based on ancestry. So these are self-reported African Americans, and found that those African Americans that had a higher proportion of West African ancestry actually did respond to the drug, and it could have been beneficial and helpful for them. And so that's what I mean by leveraging ancestry to move us towards this individualized medicine or precision medicine. It's taking us one step further in that direction.
That's great. Yeah, I think you hit the nail on the head with the evolution of precision medicine and how important this will be towards that. So with the development of tools such as 23andMe, have you found that individuals are more curious about their genetic testing and attitudes? And did you find in your study that this differed between races or locations?
Yeah, it does. It does. There's a lot more people that are interested and they get excited about, you know, this genetic testing for ancestry. And it does differ across groups. You know, some groups, like for instance, many European communities aren't necessarily that excited about, or they don't value it as important as, let's say, African Americans. And because they already know, most Europeans already know their family history, their family structure. They just want sort of to confirm it, or they want to, you know, in many cases, they may not know one side of the family or whatever. But for African Americans, it goes beyond just the immediate family structure. This goes back to being a descendant of an enslaved African that was brought here. You have no idea where they came from. And so there is much greater importance and value on genetic testing among African Americans than it is for other groups. And there are other groups out there that are very similar where, you know, maybe they've been isolated, or they've been they were, you know, some war that occurred or whatever, where groups had to move out, you know, so there are some pockets of communities like that. But for the most part, African Americans value this a lot differently than other groups. And it's because of the transatlantic slave trade and being cut off from your ancestral sort of homeland.
Yeah, I think the maps that you had included in your presentation really hit home with that. So I think that was displayed very well. Another question we had is actually a part compliment, part question. This participant said, "You're incredibly productive in disseminating your findings in scientific journals and popular media. How has that informed your and your colleagues' research, perhaps participant recruitment?"
Well, obviously it helps with recruitment because people know about your work, and they get excited about it when, you know, because of the fact that it's disseminated across many different media. I tell my students all the time, the best scientists are actually the best storytellers. And if you can get folk excited about what you're doing, if you can get people to buy into this, you're going to get a lot more support, and it'll be a lot more exciting and easier for you as you go out and recruit or get participants or even get students. I mean, so part of this for me has been one of the things I've learned is if you're a really good storyteller and you can get folks excited and want to buy into your research, it does help. But I'll be honest with you, early on, it wasn't easy to get a lot of what I was saying published because it was contrary to what the status quo was at the time. And I'm talking about early 2000 when we first started working on ancestry. There was a lot of pushback among the epidemiologists because they felt that it was more work. or it wasn't going to be that informative. But now, as I showed you, I mean, we can go marker by marker across these chromosomes and look at the rich mosaic of ancestry, and then also say something about different phenotypes or disease risk or outcomes based on those particular blocks of ancestry. So this is a very exciting and fascinating time right now. And I really want to see how we can integrate all of this with the behavioral stuff. And also, you know, what's the utility with race? I mean, I don't want to throw race out of this discussion, but I do want to make sure we don't oversell what race does. And it's just a crude proxy.
Right. I think there's a lot of exciting potential in this research. So thank you again for presenting. Looks like we're unfortunately out of time for questions, but thank you for being here today.
Thank you. Thank you all for inviting me. Had a great time. Thanks.
Dr. Christine Hunter, would you like to introduce the next speaker?
I would, thank you. It doesn't look like I can start my video, but you've seen what I look like, so I'll just move along. Thank you, Dr. Kittles, for just a wonderful and engaging presentation with a lot of important points. It's my pleasure to introduce Dr. Nancy Schoenberg. Dr. Schoenberg is the Marianne Pearsall Professor of Behavioral Science, Associate Vice President for Research on Health Disparities, and Director of Center for Health Equity Transformation at the University of Kentucky College of Medicine. Her talk is titled Poking and Prying with a Purpose, Community Engaged Research to Achieve Health Equity. Thank you, and I'll turn it over to you, Dr. Schoenberg.
Thank you, Dr. Hunter. It's a pleasure to be here, and I want to express my appreciation to OBSSR and to all of you today and to my colleagues. And I also want to pay homage to one of my disciplinary ancestors, Zora Neale Hurston, who was so much more than an American folklorist. Zora Neale Hurston was an ethnographer, a scholar, an ethnomusicologist, a performer. And she said, research is formalized curiosity. It is poking and prying with a purpose. Today, I'd like to share with you the purpose of much of our work, which is trying to achieve health equity through community engaged research. Next, please. So what I'll be sharing with you today are my disciplinary backgrounds, the anthropological contributions that really have shaped my work in the community, and that married with the evidence-based health equity interventions that so many of my colleagues who have worked with OBSSR and NIH in general have helped us to develop. All throughout this talk, I want to also tell some stories, as Dr. Kittles has suggested that we should. Tell some stories that come from my field work, which in anthropology is what we call our laboratory. In my field setting, in Appalachian, Kentucky, and rural America, you'll see a lot of photos today from Appalachia. And many of these were taken by my project manager, Tiffany Scott, as well as other people who have been involved with my work. Next, please. So the culture concept is a tricky one, and we anthropologists understand culture as sort of our calling card. It is a shared framework that is internalized in any one particular group. It is a lens through which these group members understand their reality. It is also a doing and being thing. It involves tools and processes through which group members make sense of their world. These tools are language, their religion, their kinship patterns, and their regional identity. Next, please. It is, despite its great utility and how it informs all of what we do, it is a very slippery and challenging entity to understand. As Bill Dressler indicates right here, it informs every single aspect of human behavior, but in no other variable that we in health research look at is as poorly defined or as poorly tested as is the culture concept. And Margie Kagawa-Singer went a step further, and she said, culture is oftentimes the residual variable once researchers sort of account for everything else and they can't come up with any other associations. Oftentimes people will shrug and say, well, it must be culture. So with the wisdom of OBSSR and the NIH, Bill Elwood and Marjorie Kagawa Singer and Bill Dressler and Sheba George convened a panel of 25 social scientists to help our fellow behavioral and social scientists and other scientists operationalize culture. That is what you see here, a monograph that is available for your edification on the OBSSR website. It's very, very useful. Next slide, please. So what makes the culture concept so difficult is that most culture is hidden beneath the surface. Surface culture is what we oftentimes grapple with and understand. It is the external manifestation of this blueprint for life. It is food and dress and language. But what really informs our health behavior, really shapes it, is all the stuff that's lying deep within culture. Next, please. Another entity, another construct that we draw heavily on in anthropology is called cultural relativism. It's what you learn when you take Anthropology 101. It is really the ability for us to understand a culture on its own terms, rather than making judgments using the standards of one's own culture. It involves a number of different principles. It assumes that no one culture is superior to another, and that in fact, when we understand the culture within its context, then we can start to understand why people do what they do. So here's a story from our own field site. So as we were coming around the corner of a holler, an Appalachian holler, which is like a neighborhood, we found this old truck. And this old truck, to an outsider, looks like something rotting away, some piece of trash that someone haphazardly just threw out there. And in fact, it could be a venue for tetanus. It could be something very challenging and very problematic. And the outsider might say, why don't these people, quote unquote, take care of their trash and put it where it should go? Now, an insider might have a different interpretation, and to get to understanding that insider interpretation, you'd have to ask them. And what they might say is, well, this was Papa's first truck, first vehicle ever. And he bought that on the basis of his hard-earned money, that where he went down into the coal mines, and he was so proud of that truck, and it's a living legacy to him. Another person might say, another insider might say, well, there are lots of salvageable parts in that, and it's sort of an economic security. Maybe we'll get the whole thing up and running. And yet another person might say, well, if you know this area, you would know that getting rid of this thing is no easy bargain. There's no curbside trash pickup. There's no recycling. So we just keep it where it is. Next slide, please. Cultural humility is another construct that has been gaining increasing visibility in the world of cultural competence and replacing cultural consonants and so on. It is simply the idea that every person has something of great value to learn from everyone else if they continue to reflect very critically, and if they recognize that there are power differentials and hold institutions accountable. Next slide, please. So here we are talking and trying to get feedback and trying to get input from the community. Next. Another anthropological contribution that is increasingly recognized in public health and in health and behavioral and social sciences is a holism that defines health behavior and health outcomes. The NIMHD research framework focused on these various levels of influence, from the individual all the way up to societal, these domains of influence from the biological and genetic, as Dr. Kittles just told us about, all the way up to the healthcare system. It is very important for us to collaborate, and I think increasingly we are, on addressing health and understanding health by looking at this entire complex continuum of the influences and the domains that impact health. Next, please. Also complex, like culture, is Appalachia itself. So I want to talk to you a little bit about where we do our work. And a lot of these same comments apply to rural US overall. Appalachia defies easy characterization because it is 420 counties that goes from New York all the way down to Mississippi, 13 states, 25 million people, or about 8% of the US population. It's a geopolitical designation that you can see takes up a large swath of our country. Now, when you think of Appalachia, you could probably think of the yellow and the orange areas that you could see outlined here. And you might also think of this beautiful landscape. But, next slide, please. What you may also think of is the severe disparities that Appalachian Kentuckians and rural residents experience overall. In our state, we have the unfortunate distinction of experiencing the highest rates of cancer deaths, pretty much that same picture for substance abuse deaths and chronic disease overall. In fact, compared to the US in general, mortality is about 5% higher and 18% higher in years 1990, 92, and 2009, 2013. Compared to national average. So thinking back on that NIMHD framework, we know that social determinants of health, some of the social determinants of health account for a lot of these inequities. Issues like poverty, unemployment, lack of access to social and to healthcare services. They intermingle with behavioral risk factors like smoking and substance abuse and suboptimal energy balance. Next, please. So much so that looking at the premature mortality in Appalachia, we find that, in fact, over the past 20, 30, 40 years, we have a rising and increasing rate of premature mortality. And this is really in very sharp contrast to the United States as a whole. So years of life lost before age 75. Fortunately, we see huge declines in the United States overall as a nation, but in the Appalachian context, we see increases. And I'd like to point out that these increases are not only due to substance abuse or the opioid epidemic. That certainly is a contributor, but this started happening in the 1980s, long before opioid epidemic was such a challenge in the Appalachian context. Next, please. So what accounts for these really challenging changes in the Appalachian community? Well, my colleagues and I suspect that there is a healthy migration issue going on here. So while the rest of Kentucky, as you can see here on the graph on the right, the rest of Kentucky and the US, they are increasing in their population. Eastern Kentucky is decreasing in its population. So we suspect that a lot of the healthier people are looking for new opportunities and they're migrating out of Eastern Kentucky or Appalachian Kentucky. And that means that a lot of people who stay in Eastern Kentucky, although certainly not all, as you'll see in just a moment, a lot of people are older and they're sicker. Now, why is this happening? Well, if you go back to the holism, that's a calling card of my discipline of anthropology, you'll know that the economic basis for this area, for this region, is an extractive economy that has been based for the past 150 or so years on many companies coming in and extracting natural resources and leaving the population oftentimes bereft of important resources that they can use to benefit their lives. An example of this is dwindling coal jobs. So starting in the early 2000s, we saw a huge decrease in coal mining jobs and coal in general. In 2016, there was the promise of increasing coal and coal activity, but that has actually not happened at all. In fact, in the place that the county in which we are located, in Letcher County, in 2011, there were 40 active mines, and now in 2020, there are only 8 active coal mines. So you can imagine what this does to people's viability and remaining in the community. Next slide, please. So we in health behavior and health research, we tend to focus on some of the challenges that our communities face because we're researchers and we want to solve problems. But it's really important to focus also on the assets and the strengths of local communities. And in the Appalachian context and in other places, of course, there are numerous assets. Look at this place. It is beautiful. And even more beautiful, next slide, please, are the human assets. And those are the people who comprise our work and who allow us to do the work that we do today. So I want to now spend the rest of my time talking about a couple of examples from our own work. And the first one will be a project that we did on cervical cancer that's really started the ball rolling for us. Next slide, please. So in community forums like the one that you just saw, we talked to community members and tried to gauge what they're most concerned about within the health of their community. And during some of those forums, we found out that cervical cancer constitutes a huge concern that weighs on people's minds. People's grandmothers, mothers, sisters, next door neighbors, wives were dying from cervical cancer in rates that really were unacceptable for a lot of different reasons, most notably because no one, of course, has to really die from cervical cancer anymore. It is pretty much a preventable condition given Pap tests and now HPV vaccinations. But yet, if you look at this map, which we used when we were starting our projects, you will see that the spine of red is the Appalachian region, and the reddest of the red is Eastern Kentucky and West Virginia. We had the highest rates of invasive cervical cancer in the nation, pretty much. 67% excess above the national average for invasive cervical cancer incidence. Next, please. So what did we do? This is a pattern of our work that you will see. I will describe throughout our presentation. We asked the question, what would work here? How can we address this problem here in the local context? How can we use evidence from what you all, the scientists who are out there, have provided and meld that evidence with local adaptations, understanding the culture, understanding cultural relativism, and employing a lot of cultural humility to get us to where we need to be? And so what we found in the literature that appealed to people and appealed to us was focusing on lay health advisors or using lay health advisors as our sort of vehicle to encourage women to get Pap tests. And so we sought out the input from the community through community interviews, through focus groups, through a number of different orientations. And we found that actually people would prefer non-healthcare professionals, which was good because if you think about our local context, we have severe health professional shortages. So to draw on local talent, and these ladies are emblematic of local talent, to get trained and to be those promotoras would be very, very helpful. And that's the model that we followed. In addition, we knew that we'd have to locate these interventions within a sustainable entity or organization. And people told us that one of those organizations was the church. And so we have located many of our projects now in faith-based partnerships. And they also said that going beyond the disease, making sure that you're asking beyond just about Pap tests and why don't you get a Pap test and here's why you need to get a Pap test, and going beyond that and trying to move toward what makes it difficult for you to get a Pap test and how can we help get you there. If it's transportation or if it's a lack of social support, that's where we need to go. So what we did was we trained these ladies and to be these promoters of motivational interviewing. We held group sessions and we located them within churches and other faith-based entities. Next, please. And what we found was our recruitment, we recruited about 430 women. This was a specific age group, 45 to 64, an age when Pap tests actually declined precipitously. And we found about a third of the women had never gotten tested for cervical cancer, or it had been at least 5 years since their test. And we found that our intervention really reached a high level of efficacy and effectiveness. That we found that women who were randomized to the treatment group were twice as likely to receive a Pap test, and that about a third of our participants actually did get a Pap test, and that this was sustained over the course of our project. Next, please. The National Cancer Institute found this compelling enough to administer, to place on their RTIPS website. So please, if you want some more information about this, go to the RTIPS website. Next slide, please. So always with our ear to the ground, the next project that we engaged in went beyond Pap tests and to smoking cessation. Now, I'm going to say that we were very surprised that the community members wanted us to do something with smoking cessation, because if you recall me saying extractive subsistence-based tobacco has long been a way that people in Kentucky support themselves. Tobacco was very pervasive. Family farms, people would grow tobacco to support their children's college education or to buy that new vehicle or something. And so we really had a tobacco culture in many ways, a familial and cultural acceptance of tobacco that would have made me think that smoking cessation would not be an area of focus. But, next slide, please. If you look at the average rate of smoking in the United States, and you look at Kentucky, we've got the highest rate of smoking in the nation at about 26%. And if you look at the eastern portion of our state, you will see that about 31 to 39% of our community members smoke. So people know that these are high rates, and of course they are concerned about this. So next slide, please. So we drew on our past practices for our intervention, and we located this intervention again in churches. Now, why do we use churches? We partner with churches, and not use them, but we partner with churches because they are sustainable. This church is not going anywhere. It's the Whitesburg First Methodist Church. It is going to be there. It's across the way from our project office, and it is not going anywhere. It has facilities that we can use. It has a minister who will speak on our behalf. It has a lot of outreach efforts. It's a comfortable place. People know where it is. So this has always been a very important partnership for us. There are churches everywhere in Eastern Kentucky, so this is a very accessible entity for us as well. So when we did our work, when we first started doing our work, we thought, well, gosh, this is going to be, just easy, no problem. But what we come to find out is some of our smaller and more traditional churches, actually people did not sign up for our intervention, and we were puzzled. And what we had to do was figure out why it was that our recruitment was lagging behind what we would have expected. Come to find out that for those more traditional churches, smoking is seen as a sinful activity, and people were not willing to raise their hand and say, I'll join your intervention, I'm a smoker. They continued not to do that until opinion leaders, specifically a lot of their ministers or their pastors, would say, hey, I'm a smoker, I need to sign up. And after those opinion leaders would come clean, that's when we first started getting really good returns on our intervention. So what we did with this was we, again, took an evidence-based intervention, and we said, what will work here? And so, next slide, please. We managed to take our intervention, next slide, please, and go from this evidence-based intervention to a next slide, Thank you. To another intervention where we had local health workers who implemented an evidence-based intervention that had been modified. And we went everywhere. For example, this small church. And people appreciated the social support of a small community organization like a church. They appreciated a non-professional. This intervention had been up and running in the health department, but people wanted something that was closer to home. So as you can see, the results suggest that this intervention did very well. Those intervention churches had about 30%, 32% of participants quit and remain quit. And this was a huge, huge victory for us. Next slide, please. And this is one of our project leads who was responsible for helping us break the mystery of why people were not showing up. We had about 600 people ultimately who participated in this intervention. Next slide, please. So in conclusion, we draw on our local assets and we employ evidence-based approaches that my fellow behavioral and social scientists have developed over the course of many, many decades. As the middle picture suggests, we work with a number of different denominations. In the Appalachian context, it's all Christian churches. So we wind up working with lots of Baptist denominations, Southern Baptists, Old Regular Baptists, Primitive Baptists, but other denominations as well. We work with mainline churches. We work with Pentecostal, evangelical, all sorts of non-denominational churches, Latter-day Saints, Mennonite, and so on. We go to large churches, to small churches, wherever we are welcome. We also work with our community-based clinics. And so our community to clinic navigation to improve diabetes outcome, our current diabetes project has us go to a range of community clinics, and we have lots of clinical partnerships. And then, as you can see all the way to the right, we go to homes and we go to where people are. This is our NIEHS project, or Research to Action project, where we try to address the huge, tremendous respiratory disease inequities that characterize this region. Next, please. So we've developed a process to optimize health and well-being via behavior change. We start out acknowledging culture and using cultural humility. We try to listen as best we can to community concerns, take those interventions that have an evidence-based and adapt them. We are located within the community. We have a project office. This is our project office on Main Street. We've recently relocated, and we go to where people are in their own home sites. We don't ever assume that someone will not want to join our project. We go out there and we recruit them no matter where they are. Next slide, please. So the unexpected happens all the time. Humans, as you know, are not predictable in a laboratory setting. So imagine humans within their own home setting. We try to be rigorous. We try to have transparency, and we try to have replicability. We try to do exactly what we said we were going to do in our grant applications, but sometimes the unexpected happens and we cannot control the circumstances. That happened with the churches that I described. That's happening now with COVID, as many of you know, and that happened in this picture. So in this photo, this was the project that Dr. Christine Hunter was our program officer for, an intergenerational approach to improve energy balance, an NIDDK supported project. Our kickoff day happened to be on a lovely day. It was not predicted to snow, but in fact, we got 8 inches. This is the road that was supposed to take us to the Cooperative Extension Office where we were supposed to have 150 people waiting for the kickoff event. Needless to say, this didn't happen much as we had anticipated. Next slide, please. In another project, this is our NIEHS Research to Action Mountain Air Project. We do old-fashioned enumeration. Every 5th household, we try to knock on the door and ask about respiratory disease. So what you'll see here is that this is our enumeration log describing each of the households. So my goodness, look at the double wide. So say we had enumerated, try to go back to the double wide. We couldn't do that because of the big dogs, the big bad dogs and bigger and badder dogs that would be waiting to get us if we knocked on their door. Adaptation is simply the systematic modification of an approach or an intervention to make sure that we consider language, culture, and in this case, context. Next slide, please. Adaptation is a hallmark of what we are doing today. We normally conduct our recruitment through churches, as I've mentioned, and you'll see this church. We have a health screening, and that would be a kickoff for our diabetes projects. But in these times of COVID, it is not safe to assemble in a church, so we can't do it. So to collect biometrics, it requires transportation, drive-by, and collecting biometrics creatively here. Some people don't have transportation though, so we mail the A1C Now machines, as this happy person is showing you in this picture all the way to the right. Next slide, please. Finally, sustainment is something that is really important to us. So we know that that we build our community through training our lay health advisors or community health workers. We have trained hundreds of people over the course of these 16 years. Some of these people have gone on to earn advanced degrees, to start their own health projects, to start on new paths in life. All of our resources, or as many as possible, are provided to our rural areas. For example, this graphic, our map project, our graphic design, the food that we used for our projects. And as our Mountain Air project, our NIEHS project indicates, we try to make sure we're working with local communities to implement and to have a long sustained effect. Next slide, please. So let's let Zora Neale Hurston have close to the last word when she says that there are years that ask questions and years that answer questions. Like Zora Neale Hurston herself, we meld these two. We ask questions, we answer questions, and hopefully we change lives and improve health equity. Next slide, please. So I want to acknowledge the support from the National Institutes of Health. Through the years, the NIA, the NCI, NIMHD, NIDDK, NIEHS, and now I'm pleased to say NHLBI are supporting our work. This latest project, we're really pleased. It is the first that we know of, implementation science project, that it focuses on mHealth in the increasingly used technology-infused Appalachian region. Next slide, please. And finally, an acknowledgement and a shout out to our staff who work arduously to improve health equity. We've had many of these staff people work with us for many years. The lady on my left-hand side is our longest serving lay health advisor, Becky Cornett, and she's been with us for about 16 years. Next slide, please. I want to just throw out a call to everyone to come to visit us. We are collaborative, we're cross-cultural, we want to work with you, and we want to improve health equity. Thank you.
Thank you so much, Dr. Nancy Schoenberg. I thought that was really interesting to hear about your tailored approach to health interventions and your findings on the impact of not only trusted partnerships, but also individual key opinion leaders and health interventions. And another participant also commented that you gave a very clear explanation of the enigma of defining culture and the benefit of opinion leaders and the value of truly meeting people where they are. So thanks again for that. We do have a few questions for you. First, starting off, generalizability of findings are highly valued in scientific research, but community-based research findings appear to have limits to their generalizability. How do you determine which findings are community specific versus generalizable to other communities? And how do you compromise between a tailored approach to specific communities versus a broad messaging approach?
It's a very, very good question. It's difficult. I mean, we start off with the community asking what they want, and then we move on to look at some of the epidemiological correlates. We talk a lot to other people, especially other health equity researchers, and even though there are specific concerns that are community focused, we find that a lot of the same challenges that exist, for example, in our work in Appalachian Kentucky, also exist in some of the remote rural regions for Native American reservations and in the inner cities. So a lot of the resource deprivation issues, for example, or the transportation issues, we can see that a lot of those are happening across cultures. So what we try to do is we try to use some of those large frameworks. For example, the NIMHD framework, to get a baseline and a theoretical understanding of what are challenges across these populations, and we see what will fit specifically to that. Every place is unique in many ways, but there are large similarities that can be addressed.
Great, thank you for that insight. Another question is saying that your studies focus on delivering healthcare access to where people are. What are some of the other challenges to improve health behaviors to people in Appalachia area? For example, could the high rate of cervical cancer potentially be due to vaccine hesitancy?
Actually, interestingly enough, vaccine hesitancy is not a large issue for us in Eastern Kentucky. We're very, I guess you would say, compliant with vaccines in Eastern Kentucky and have been thus far. I think Francis Collins really talked very importantly about how behavior is going to be what gets us through COVID and behavioral studies, and how we really need to make sure that everybody's on board with the vaccine behaviorally, and that will be something that my colleagues and I will have to address. So that actually is not as much, the vaccine and vaccine hesitancy is not as much of a problem, but there are lots of other concerns. So, you know, social norms and social practices tend to come together. And given that communities tend to be tight-knit, you see the proliferation, and I think some of the social networking studies have really pointed this out. You see the proliferation of both optimal and suboptimal behaviors in tight-knit communities. So smoking, weight gain tends to be contagious, as Christakis and Fowler have pointed out many times before. And on the flip side of things, once you start behavior change, it also can be contagious. And we see this with some of our studies. We see that once one behavior starts to change, another can start to change. So addressing one vexing problem can lead to addressing others. And we hope to have this positive cascade.
Wonderful, thanks for that response. I think we only have time for one more question. The last question is, does cultural adaptation apply only in health issues such as smoking cessation, recruitment, and retention? If not, how does culture inform intervention and questionnaire design and analysis?
Oh, this is a very, very big question. So it informs everything that we do, everything that everybody should be doing. So I want to again reference the cultural framework, cultural framework for health, the OBSSR. And in that document, you will see that it sort of takes you through step by step a process for integrating culture and the culture concept into your work. It informs every questionnaire. We do lots and lots of pre-tests and foundational work to make sure that we're semantically appropriate. They're not beyond just simply language, that we're topically appropriate, that we're addressing every single facet of life and of existence. We involve a lot of partnerships And we always have people who are commenting on and running all of our projects along with me and with our team. So I think those partnerships become the element that's really, that provides that authenticity and that true cultural response.
Great. Yeah, I couldn't agree with you more. Well, unfortunately, it looks like we're out of time for questions. So Dr. Nancy Schoenberg, thank you again for such a great presentation.
Thank you for having me.
And thanks everyone for your attention so far. It's now time for a 5-minute break. We'll be returning at 2:25 to hear a presentation from Dr. Esther Duflo on translating preventative health behavior research to policy, and also an update from the director of OBSSR, Bill Riley, on the state of behavioral and social science at NIH. See you back here at 2:25.
Welcome back from the short break. It's my pleasure to introduce Dr. Esther Duflo. Dr. Duflo is the Abdul Latif Jameel Professor of Poverty Alleviation and Development Economics at the Massachusetts Institute for Technology. Her talk is titled From Research to Policy in Normal and Troubled Times, Evidence from Preventive Health Behavior. Thank you, Dr. Duflo, and I'll turn it over to you.
Thank you very much, and thank you very much for inviting me to this festival. It's really a great occasion to showcase some of our work, and in particular to thank the NIH for the support that work has gotten over the years. So a few years ago, I co-founded, along with Abhijit Banerjee and Rachel Glenister, a network called the JPAL, or the Abdul Latif Jameel Poverty Action Lab. The objective of JPAL was from the beginning the idea that we could improve lives, and in particular the lives of people living in extreme poverty all around the world with better evidence. So we started with the very simple idea that policy would be more effective, and in particular anti-poverty policy would be more effective if it was based on scientific evidence. So our main research tool is randomized controlled trials. And from the very beginning, our objective in creating JPAL was to do three things: to conduct randomized controlled trials, to train others, both PhD students, but also policymakers, in how to conduct randomized controlled trials of policies in the field, as well as understand the result and be educated consumer of this type of research. And finally, to promote the adoption of both the method as large as a tool of governance and also the results and to allow for the scale up of effective policy as well as the scale down of ineffective policy. So when we started in 2003, we were a staff of 3 people, and we had conceived of us as a network, but the network had only 8 faculty. Today, the staff is a staff of 400 people around the world with an office in every continent, supporting a network of around 500 affiliates and invited researchers who are together conducting, have either completed or in the process of conducting a little over 1,000 projects. The projects are all around the map. I'm going to be talking about health today, but they are all around the map in education, in governance, in health, in energy, and in climate. But really, the objective of JPAL was always to go beyond the research, because in fact, when we created JPAL, we thought, well, we can do the research, but what we cannot do, what as academic we're really not trained to do and not particularly skillful at doing, is to make sure that this evidence finds its way into policy. And the objective in starting JPAL is to do this translation. And I suppose a little over 15 years after our starts, we were effective in the measure of how many people were reached. So if we count directly the people who were affected by policies that a member of our network has found effective, that's about 400 million people. In addition, there are also policy effectiveness that doesn't go through scale up, but go through a change in conversations or things like that. So in that sense, JPAL has been a force a small force, just one of the forces that have led to the adoption of more effective anti-poverty policies, and have played a small role in ensuring progress, for example, against malaria, progress in childhood immunization, and the fact that there are more kids in school learning more and things like that, which is really one of the big progress that happened in the world over the last several decades. So if we can go to the next slide, and then the next one again, we can, I'll start by presenting you the sort of the straw man about how policy influence works. How do you go from research to policy? And I should say that this is not a straw man that I took completely by accident, because in a sense, this is what I was thinking when I was just starting running randomized control trial as a junior faculty. I kind of imagined this to be the cycle that this would follow. So the idea is that you run a good experiment, and to run it good, maybe it's not very big, it's very well controlled. You get interesting results. You get your paper published in a top journal, or at least in a peer-reviewed journals, and then you prepare a policy brief to start showing it around to policymakers, and policymakers get seduced by the evidence, which is very clear, and you get full-scale adoption. So this particular straw man has been criticized by various people. If your experiment is small, then maybe it is gold-plated, maybe it's not replicable on a larger scale. Also because if the samples are too small, you don't get results that are very precise or that are externally valid. And then your results, they are only valid in one place. They might not replicate elsewhere. They might not even be internally valid, because in real experiments, as opposed to, that are conducted in the field, as opposed to the very clean randomized control trials you're getting for testing a drug, for example, you get all sorts of issues. You lose people to follow up, people are not doing what you're asking them to do, are not complying to the treatment, and so on and so forth. There are spillovers between treated and control people, so you don't even know exactly who was treated. So for all of these reasons, the results may not even be internally valid. And then but even if you do have results that are internally valid, and you think that you can make the case that there is some external validity, they might not be of interest to anybody, because really policymakers have their own priorities, and they will only seize of your results if it can justify what they wanted to do anyway. And if not, then it's really not going to go anywhere. And then if, despite all of this, you get full-scale adaptation, then it might not work as well as you hope, because once you do something at scale, there are equilibrium effects. So for example, if everybody goes to school, the returns to school might go down. And so you have lower effect than you were hoping. You might have political reaction that are backlash against a politic when it's implemented at scale, or on the opposite, wide-scale adoption. For all of these reasons, the effect that you're getting on a smaller scale do not apply on a larger scale. So next slides, please. In a way, I don't disagree with I disagree with many of those criticisms, but I don't disagree with all of them. But the big way in which I disagree is that really it's not the way that policy influence works. You don't go from your small experiment and then write a policy brief and go from there to wide-scale adoption. And instead of telling you how it works in general, I thought that I would give you two examples of how the full cycle from one particular idea to adoption takes place over the years in one particular context for normal times. And then just, to make this relevant to our very special times, show you our accelerated timelines in COVID times. So not only COVID vaccines, but social science research also gets accelerated in normal times, in these troubled times. So let me start, if you want to go to the next slide and maybe the slide after that, how we illustrate how we go from research to policy in normal times. So I'll take an example, which is something I've been working on and off during my entire career. This was one of my first projects, and it's something that I've always come back to, which is childhood immunization. So I don't need to persuade you in this particular audience that vaccination is important. Lately, we are hearing a lot about the role of vaccination, but really, even in the absence of COVID, vaccination are one of the most cost-effective ways to save children's lives. And there has been progress over the last several decades on vaccination coverage, but over the last few years, this progress have been stalling. Global vaccination coverage remains at 85%, with no significant change during the past few years. And the WHO estimates that an additional 1.5 million deaths every year could be avoided if global immunization coverage improves. So to put this on perspective, this is of an order of magnitude which is similar to the number of COVID-19 deaths to date, but that's every year. And we hope and expect that the COVID-19 deaths are going to recede, whereas these deaths have been happening year on year on year. And these are vaccinations that we have. For example, about 20 million children under the age of 1 are yet to receive their third dose of DTP vaccine. And 60% of those children will also not receive measles typically. These children live mostly in 10 countries, including India, which is the setup of this study. India is an interesting country in this regard because it's fairly rich. You can't say that it's because of massive dysfunction in the economy at large, but it really has been struggling in getting preventive medicine and in particular immunization up to snuff, in particular in the north of India. So in 2010, we published the first study on the supply and demand factor on immunization. So if I'll show you, the next slide will show you after the little church in the previous call, a little health center in India. And the characteristic of this little health center is that it's closed. And it's not an accident because this particular health center, we photographed for 1 year every week and we found it closed every single day. And it's not because there is no nurse allocated to it. There is a nurse, she just isn't going there typically. So one of the problems clearly with immunization is supply. The immunization services are not regular, in particular closer to where people live in rural areas. Another issue is demand, which is that even when the services are available, people aren't going regularly. And yet, I'll show you some data in a moment that suggests it's not about defiance. It's not about people not trusting the medicine, because a lot of these parents will very happily tell you that they would love to immunize their kids. And in fact, they start the immunization course. They get BCG at birth almost systematically. And then they get, maybe one shot of DTP, maybe the second one, and then they kind of stop. And there is a big drop-off in the third dose of DTP and then in measles vaccine. So we were interested in knowing whether fixing the supply, which really has been the main response, both from governments and from institutions like Gavi or the Gates Foundation, is trying to get the immunization as close to people as possible, which is very hard because the closest it gets to people, the hardest it is to monitor. But we wanted to try to say, well, suppose you could do it. Like suppose God gives you a perfectly run immunization camp. How much does that get you? And then suppose that on top of that, you're doing a small inducement for people to take the last step. So think of the immunization infrastructure as being the last mile problem and add to that sort of the last meter problem of people actually taking the time. So if we go to the next slide, you can see a map with our experimental design, which is the villages in Rajasthan, a tribal area in the north of India, which you may have visited for touristic reason. but it is extremely poor, and at the baseline of our study had vaccination rates of about 5% for full immunization. And we worked with an NGO called Seva Mandir, who in turn worked with the government to take charge of the immunization services in some villages. And we picked 60 randomly selected villages out of 120. And in those 60 villages, they set in place monthly immunization camp that happened absolutely on the dot. So they completely solved the kind of supply problem of closed subcenter that you see in the previous slide. And then on top of that, so these are the camps, the blue villages, the white being the status quo. And then on top of that, we had a second treatment, which is they had a camp. And in addition, we provided a small incentive for parents who brought their kids to get immunized. Next slide, please. And you can see the incentives are a kilo of lentils as for each shot, plus a set of plates for completing the immunization. So not something that is very large at all, a small incentive, but it's sufficient, we thought, to give people to sort of compensate the small cost of actually taking the kid to immunization, and maybe served as a reminder that it might work out. So this program got implemented by the NGO, and we monitored immunization rate by interviewing parents as carefully as possible. And we got the results, which you can see in the next slide, which is that, so at baseline, immunization was very, very, very low in our villages, but 1% in the control villages. By end line, it had increased to 6%. But in the places where the camps were put in place, it had almost tripled to 17%. If in addition you add incentives, full immunization rate was now 38%. So another almost doubling over just doing the camp. So this is a very large effect for a packet of lentils. And what is interesting, which you can see in the next slide, is that if you calculate how much it costs you to fully immunize a child, next slide, please. You actually see that it turns out to be cheaper when you have incentives than when you don't have incentive, even counting the incentive as a cost. Although you could argue that it's not really a cost because it's lentils that go to the family, and that's a good thing. But even if you count it as a cost, it turns out to be cheaper per immunization. Why? Well, because the cost of running the camp and bringing the vaccines and all that are fixed costs, and you need to pay the nurse regardless how many kids she immunized. And with this program, you had many more kids coming, both from the village that was targeted and from neighboring villages, so that the cost per child immunized declined. So this was the study, and it got published, and it got a certain amount of coverage, and I was incredibly proud of it. I still am. This is like one of the, most clear-cut things I've done in terms of something really successful, very easy to implement, should be scaled up. So the next question, of course, is what happened to scale up? This is something which I took very much to heart and was trying to get to scale up. So we can go to the next slide and the slide after. And the point is not very much. For a long time, nothing happened. So why was that? Well, in part because that was a little bit this criticism that I was leveling against our own work, that that if it doesn't gel with the policy agenda, it is very difficult to make progress. We sort of hit that problem, which is for some reason, even though the Indian health system is full of incentives, everybody gets incentive to do everything, to get sterilized, to deliver in hospital. There was this big opposition to get incentives for immunization, which I always found a little strange, because since immunization has a huge externalities, you would think that it would make sense to give people incentive to immunization. In fact, in our countries, we just make it mandatory for kids to be immunized if they want to go to school. But this is not the case in India, and this is not something that somehow politically resonated. So nothing happened for a very long time. And then you can go to the next slide. We we were able to work with the government of Haryana, which is one of the state in which Delhi sits, one of the very large state in the north of India, which has a very low immunization rate. And they had been doing a lot of effort to improve the supply of immunization, and their immunization rate got stuck around 40%. And they were a bit desperate about, what can we do? And they got, and we got in touch with them through UNICEF, actually. And then started talking to them. And they were not particularly interested in incentive, especially interested in incentive, but they wanted to find ways that they could use to increase the demand for immunization. They realized that that was the problem. They thought there might be a core belief issue, and that's hard to affect. But again, many people we interviewed, about 94% of the parents we interviewed at the beginning of the project think immunization is very useful and that they want to do it. So that's not the only problem that there is. So what else can we do? Well, one possibility, of course, is small incentive, because maybe what happens is people tend to procrastinate and always postpone the immunization to the next time. The incentive kind of counteracts this tendency, and the pilot in Rajasthan was a good sign that this might be the case. But there was also other ideas that had appeared in the meantime. One is the idea of increase the salience of immunization by sending people reminders, which have become, of course, very common practice everywhere. The other is to use the social network, and in particular to identify ambassadors in the community that could relay the immunization message. So there, the challenge is to easily and cheaply find people who are influential in the social network and try to recruit them to become the immunization ambassadors and see whether that would work. So here, what we did when we started working with the government, next slide, is that we didn't scale up immunization. We started working with them at scale. This is now not a small pilot. This is a RCT that took place in 7 districts. We started by introducing tablets to nurses with a very simple e-health application that helped them track immunization. And that was done everywhere in about 2,400 villages. And then on top of that, we introduced various versions of 3 experiments. One is incentives again, where lentils were replaced by cell phone minutes, which are easier to administer. One is targeted SMS reminders, and one is this mobilization of the social network. So how did the mobilization of the social network work? We went to the community, interviewed 5 people at random, and actually 15 people at random, and asked them, hey, who do you think is an influential person in your community? Just that simple. And it was actually, they gave us some names, and they tend to give us all the same names. So people tend to agree on who are like the gossip or the people who talk a lot in the community. And then we went to see them, and we asked them if they wanted to be ambassador for the program. And the good things about them being the village gossips is that they like to talk, so they were very willing to do that. What is interesting is that this was always meant to be a preparation for policy. So it had a number of ingredients. The first one is the scale, because we wanted to have all of the problems that you can have if you run something at scale. The second one is that we had lots of options, because we have these 3 experiments which have various combination possible and various doses. So in combination, it's an extremely complex design, which I'll show you in a minute. The idea was not complexity for the sake of it. The idea is that out of this, we could really identify the best option for the government to scale up. And then in addition, we want to tell them where it would be most effective to scale up. So we combine this with a machine learning algorithm that can tell them where which program works the best. Just to give you a sense of the timeline, this takes a long time. Our discussion started in 2013. The full design wasn't elaborated till 2016. Next slide. Next and next slide. That's the beginning. 2013 we started, and then that's the continuation. And finally, in 2018 we got to start our experiment. So I compressed the timeline when I described to you how it works. So in the next line, you can see a map of the area with the treatment and control PHC in one particular district. We had 7 such districts, just to give you an idea of how the randomization worked. And then the next slide has the experimental design chart that gives you a sense of all the combination of treatment that exists. So first we had village with and without incentives, and then various level of incentives. Then we had village with and without SMS, and then various level of SMS. And then we had village with and without ambassadors. And if we had ambassadors, we selected them in various ways. Just a note on implementation. This is so next slide and next slide. Before we did any of that, we had to work with the government to have a correct MIS to record all of the identification. They actually didn't have an e-health system at the time, so we developed one for them with a team of data scientists specializing in health application at MIT. So this was a very simple tablet application that the nurses used to register the kid and then collect the immunization that could serve as a backbone for both for us for the data collection and to administer the program. You can see in the next slide the number of kids that were immunized. In total, over the course of this project, we had almost 300,000 unique kids, and 74,000 of them got fully immunized. Next slide, you can see that on top of the, because we are going to be relying on administrative data to evaluate the program, we had to check that this data was good enough. So on top of it, on top of the administrative data, we wanted to make sure that there was no manipulation by nurses, for example. So we sent teams to randomly check both that the immunization session were happening on time, and then to interview in each village a few parents and check the report of the parents with the records that we are getting from the platform. So, for example, we know whether the kid exists, and then if they exist, whether they in fact have been immunized and for what. And fortunately, we were very lucky that the nurses took their job super seriously and they do this well, and the administrative data is of great quality. It's actually much better than the data that we used by interviewing the parents in the Rajasthan pilot. So in the next slide, you can see our primary outcomes is simply the number of kids that are fully immunized every month in the village, which we proxy for the purpose of analysis by the number of kids who got measles, because measles is the last vaccine in the sequence. And if a child arrives and they don't have measles, they give them the DPT, for example, at the same time. So we could have constructed a fully immunized kid, but that's almost equivalent to measles. So that's one outcome. The second primary outcome is the cost per kid immunized. How much, because it could be that it's not the same policy that is most effective and most cost effective, because a very cheap policy could be less effective but more cost effective. In the next slide, you can see the sort of average result, just comparing each intervention to each other. So these are the treatment effect for various interventions without worrying about how they interact to each other as yet. And what you can see from this is that incentives, as in the Udaipur work, incentives are effective. Here it's the highest level of incentive that is the most effective. And it's important that they are increasing with each shot, so that people get more and more as we go along. In the first project, we had them constant, but we gave a plate at the end. So we were interested in knowing whether that was important to keep that increasing schedule. And it seems that it was important to keep motivation. SMS by themselves proved to be disappointing. They kind of have no effect on their own. On the other hand, the gossip experiment, the idea of having immunization ambassadors that are reminded by SMS, in fact, to remind their friends to go to the immunization camp, was almost as effective as the most expensive incentives. And it's, of course, much cheaper to do. So this was very encouraging that this was kind of the new innovation in this particular experiment. Even though I really like incentives, I'm very happy to find something else that works even better and for a lower cost. So the first analytical exercise that we are doing is then to see, well, out of all of this policy combination, which is the one that is working the best? So that's in the next slide. Now, what do you do? What do you need to do? What gives you the biggest increase in immunization? And then we get a result that maybe is not particularly surprising, but it's to do everything. That you should combine incentives, gossip, and SMS together. And in fact, the different policies really reinforce each other and have strong complementarity effect. In particular, for example, because the gossip can remind people that there is an incentive, and therefore they are more likely to go. And they can remind people that it's fine and immunizations are good, and that it's not to fool you that they give you an incentive. So doing all three together is the most cost effective. It's the most effective, sorry. However, contrary to Rajasthan, the only cost-effective policy in the sense that, next slide, the only policy that reduce the cost per immunization compared to the control the group, compared to doing nothing, is the combination of immunization ambassadors and SMS. So why is the incentive not effective in the not cost effective, even though it's the most effective? It's because you have to pay the incentive to everyone. And in this case, the immunization levels are higher than what we had in Udaipur. So you end up paying incentive to a lot of people who would have done it anyway. So that ends up being quite expensive per additional kid vaccinated. Whereas the gossips and the SMS are so cheap as intervention that given that they are almost as effective, they become much more cost effective. So next slide. So what's the policy advice? Remember, we started this discussion with the government of Haryana. They had little academic interest in the study. They really want to know what they should do. And so our first piece of advice is that they can use gossip plus SMS everywhere. And in fact, just before COVID-19 hit, we were working with them to scale it up. And now we are going to start this again. And then the second question is that, well, but it's sad to give up on the most effective policy because it's not cost effective. So are there places where it would be more, it would also be cost effective? Can we identify some places where it would be cost effective to do this most effective policy? So there, what we did in the next slide is to develop an algorithm, a machine learning technique, along with some statistician in my department, to look at how to identify in a principled way, without avoiding cherry fitting, in which places the program is the most effective. And what you're finding in the next slide is that there is a ton of predictable heterogeneity in the treatment effect. So what each of these bar and whiskers show is a treatment effect by quintile of predicted effectiveness. And you can see that the effects are very high in the places where it's most effective, but they're actually negative in the places where it's least effective. That means that there are places where this combined intervention actually hurts. So it's very important to know, because you don't want to do it there. And you want, if you're going to do it anywhere, you want to do it in these places, in these villages in the top quintile where it's the most effective. And where is that? Well, you can kind of characterize these places. You can go to the next slide. And maybe that's not going to surprise you, but it turns out that it's in the places where immunization levels were the lowest. the immunization levels are much lower in the in the most affected villages than in the places where it's not the most affected. That might seem totally obvious as a conclusion, but it wasn't obvious to me. I would not necessarily have bet that this is the result we are finding. And then once we're finding this result, it's actually useful because it is a nice happy coincidence where equity and efficiency coincide, because you can go and help the most in the places that need it the most. And it's also going to be where you get the biggest bang for your buck, because it turns out that in those places, it is cost effective to run this full intervention. So our sort of final policy package, then, next slide, is the combination of gossips or immunization ambassador and SMS everywhere. And then to add the incentive in the places where immunization levels are very low to start with. Let me go 2 slides ahead. I only have 5 minutes left, and I want to talk to you about what happened during COVID-19. So you can do, oh yeah, perfect. So when COVID-19 hit, imagine, piloting this big ship with 500 researchers running very many experiments, and you really have to first stop it immediately and then turn it on a dime. We couldn't do any research with human subject anymore involving direct contact. We could only do things that were to do on the phone, and everything had to be done from home. So first of all, we needed to be safe and make sure that our subject and our staff was safe. And second, we needed a research that could lead to immediate policy action. So by that time, we had a few things going for us. We had experience in encouraging preventive behavior, like immunization. We had some credibility, and we had good contact with government established over the years by this long project and hanging out with governments. Also, Abhijit Banerjee was named the head of the West Bengal COVID-19 task force. So he turned out to be a little bit, although not being a medical doctor, to be a bit the equivalent of the Dr. Fauci for West Bengal. He was coordinating the team. His team was full of doctors, but he was a chairman of that committee. Still is, actually. So the first thing we did is to try to send messages. Next slide. And here the question is, at the beginning of the epidemic, there was so much information and misinformation flying around. And our question was that, can we send some messages with some basic information? Would that help? Or would that be completely drowned in the noise? So what we did is that we used our next slide. We used our own Abhijit Banerjee, who was the head of the COVID-19 task force, and who, since getting a Nobel Prize for economics that year, was a bit of a household name in West Bengal, where he comes from. And he recorded short video clips. And we sent them to people via SMS. So we sent them to 20 million people in randomly selected zip codes. We left a few zip codes as control. And then we varied various aspects of the message, but the most important thing was to know whether the message would work at all. And then we called people. We both called the health worker to find out if anybody reported symptoms, because one of the things was to encourage people to report any symptom. And then we asked people from the community to ask them about their behavior. And to our surprise, next slide, the treatment had very large effect. A simple video had very large effect, even though it was a high information environment. The symptom reporting doubled. And we also saw an improvement in behavior, interestingly, not only among the people who were directly targeted, but also among the people from the community who were not part of the network where the messages were sent, because we only had one full network covered, but who presumably were informed by their friends. So again, we're seeing the social community playing a big role. So here, the next slide, the chain from, in this instance, the chain from policy action, from research, although this was already research at scale, to scale up was much, much, much faster, much compressed. because what happened in West Bengal is that Abhijit was asked to record 2 more messages, which were then sent to millions of subscribers, not as part of an experiment. It was just a policy. So he did it twice. And the second time was to ask people to not travel en masse to Durga Puja. So that's the equivalent of Thanksgiving, if you want, plus a religious ceremony. So imagine encouraging people not to gather together in big religious ceremony. And I don't know if this is due to the experiment, but the attendance was much, much lower that year. And also there was no uptick post Durga Puja that people initially feared very much the same way that we are fearing an uptick post Thanksgiving in the US. So that was for West Bengal. And then in the US, so as we were working in West Bengal, we started working in the US as well with doctors from MGH, recording messages mainly targeted to minority, African American and Hispanic at the beginning. I'm not going to detail the results because it's still under embargo, but the bottom line is that we were confident enough that when Facebook, some people from Facebook approached us having read the West Bengal paper saying, are you interested in doing something with us? And we agreed to do with them a campaign before Thanksgiving, where doctors recorded 15 seconds promotional videos. You can see one of our doctors here on the slide, which basically said, if you love, the best way to show your love is to stay home. So very much like many doctors individually did it. And this video was sent to 40 million people in total in the US. Usually people received it more than once. Stay tuned for results, but hopefully it will have some effect. Even if it doesn't, this is the much faster scale up from the West Bengal result as well as our preliminary US-based results to immediate scale up in this world. So in conclusion, research, this is last slide. Research is more and more embedded with policy, so there is no sense in which you can really go from a small pilot directly to policy implementation, but it's become really possible to work with policymakers to make research being part of their activities, such that you can shorten the path between the research and policy action. And this path was greatly shortened during the COVID-19 crisis. So social science also plays a fundamental role in keeping people healthy. And I'm sure it's going to play a very big role, for example, in ensuring that, or in making sure that people get vaccinated, for example, against COVID once the vaccine becomes readily available. Thank you very much.
Thank you very much for your presentation, Dr. Esther Duflo. Your research definitely had an incredible impact to the communities that you studied, and also offers an excellent scope for how we can approach other health interventions, including COVID-19 vaccination, which you suggested. And we also appreciate you sharing how we can improve the transition of research into policy. So we do have time for a few questions. A question that we received is that the impact of community ambassadors in your project has been very substantial. What did you learn from working with influential people suggested and trusted by the community? Did they want to have a say in the message that was portrayed, or how was this relationship conducted?
So that's a very interesting question. So I suppose they had a lot of say because they weren't compelled to say anything in particular. I think they were very interested in talking. They were interested in talking to us about why are people not getting immunized, and then they were interested in talking to people about getting immunized. So most of the people that we approached for this program were willing to do it. And you're absolutely right that they were happy to be involved in the development of the message.
Wonderful. Thank you for that insight. Did you find that the use of incentives conflicted with autonomy in creating a long-term social norm in your study in India?
So I've never been too worried about that, the sort of the conflict between long term and short term, because at some level, to me, if it worked forever, then I think it would be perfectly fine to have incentives forever. For example, in the US, I don't think there is a discussion that we should make it very easy for people not to get immunized before sending their kids to school. So in the US, we have very strong, or in Europe, similarly, we have very strong incentives to get immunized. And nobody is really discussing removing them to create autonomy for people. This is, in a sense, immunization is something where, in particular, the closest you get to herd immunity, the smallest the private gain are, and the largest the social gain. And therefore, to me, maybe this is narrow-minded economist view, but to me, it is not, it makes perfect sense that you don't want to compensate people for doing something which is costly individually and not that useful individually, because when the disease becomes close to eradication, there is less value to do it privately, but as a huge community impact in the form of herd immunity. So I think it's very different in the case of a COVID-19 vaccine, especially the ones that we have right now, where there is very, very clear evidence of a private benefit, and we don't yet know where there is a social benefit. Then there you kind of want people to do it because it's good for them. But in the case of many of the childhood immunization, it makes sense for me that we either have a mandate or we have incentives. The question where the question becomes relevant is that I think it is possible in some contexts that incentives are counterproductive, that people interpret them as a signal that there's something wrong with what you are trying to sell them. And in that case, then they're bad, like they don't work. So that's why I think looking at the heterogeneity across contexts, like we did in Haryana, proves to be very important. And it turns out I'm very interested in doing some more work on why the incentive proved counterproductive in some of the villages. And I think it's because they were very small and people were already doing it. And they're like, oh, I was already immunizing the kids, but now you're paying me, and you're paying me so little that it really must not be that useful. So you can have a reasoning of that form.
Yeah, that's a really interesting perspective. One of our listeners was asking how one gets involved with JPAL and with researchers working on projects related to uptake of COVID-19 vaccination in India. Do you have any resources that you can provide?
Yes. So I think the best best place to start is to go to our webpage and all of I can send it to you in the chat, and then you can spread it to other.
I don't know if the audience are able to see the chat actually.
So it's www.povertyactionlab.org. And I think that's a good place to start because you're going to find the names of the people who are working in all of the sectors, as well as the contacts. We have a big office in India, in Delhi, and they would be, I'm sure, quite delighted to answer your question and see where there is ways that we can work together.
Wonderful, thank you. Well, unfortunately, it looks like we're out of time for questions. Dr. Esther Duflo, thanks again for a wonderful presentation and for joining us today.
Thank you.
And Dr. Christine Hunter, would you like to introduce our next speaker?
I would. It's my pleasure to introduce Dr. Bill Riley at the National Institutes of Health. He's the Associate Director for Behavioral and Social Science, and he's also the Director for the Office of Behavioral and Social Science Research. So, Bill, it's all yours. Thanks.
Thank you, Christine. And let me again thank Rick and Nancy and Esther for their wonderful presentations today, as well as the presenters from yesterday. I think many of us go to scientific conferences that are consistent with our areas of interest. And one of the things I particularly like about the festival each year is that it shows the breadth of behavioral and social science research that the NIH supports. I'm always excited at the end of these to just see that breadth, as well as the quality and the rigor and the commitment to health-related behavioral and social sciences research. So thank you all, much appreciated. As is our convention every year, I'm going to spend a little time talking about the state of the social and behavioral sciences at the NIH from the prior fiscal year. So let me go to the next slide. And the next slide after that, because I'm going to start with our overall behavioral and social science research funding, which, as you can see, has grown steadily over the past few years. It's grown both in basic behavioral social science research. It's also grown in the applied area as well. The increase slightly smaller than it was in previous years. We had a pretty good bump from 2018 to 2019, as you can see, but still a nice consistent increase in behavioral and social science research funding across the NIH, particularly in this last year. Next slide. What you'll see, though, is a little bit of consolidation, a slight slip in the total number of grants awarded by the NIH in the behavioral and social sciences research area over the last year. I actually think that's probably a good thing. I mean, it's clearly some more research that we need to do internally to see how this goes, but I think we'd all agree that we would prefer larger grants that are well-powered, and maybe a few less of them than more but smaller and less well-powered sort of projects. So if the result of this consolidation is that we're getting more large and well-powered projects that are being funded, then I think that's a positive for the field in general. Next slide. Everybody always asks, well, but which ICs are the ICs that seems to be doing the most? And this shifts a little bit every year. So this is for FY20, and NIA and NIMH were at close proximity in terms of the total number of behavioral and social science research grants that they funded this year, and consistently are among one of the strongest, both from the population science end of the spectrum and also from the neuroscience end of the spectrum. But of course, a lot of the other ICs, as you can see, NIDA, NICHD, NINDS, NCI, et cetera, as you go down this list, even to some of the smaller ICs that you would not typically think have much in the way of behavioral and social science research that they fund are actually doing some of that work. So, and actually for the Office for OBSSR, we see part of our value not only in assisting the large ICs with large behavioral and social science portfolios, but particularly helping the smaller ICs with smaller portfolios to leverage what they currently have and expand their capabilities by doing so. Next slide. So as you know, or some of you know, a few years ago, we wanted to get a better view of what behavioral and social science research we fund at the NIH. So we asked our colleagues in the Office of Portfolio Analysis to do some natural language processing work for us and see if we could subdivide or subcategorize and cluster the various sort of content areas of behavioral and social science research that the NIH funds. And what you see here are the 13 sort of clustered areas of research and how we describe them. So let me go to the next slide, which will show you how we funded this. And this has been pretty consistent over the years, that our primary awards in behavioral and social science at the NIH are mostly in the sort of cognitive science area of attention, learning, and memory, but also closely behind in social processes and determinants, mental health, which is a broader category. That's not just funding by the NIMH, it's actually funding for things like depression and cardiovascular disease and those types of things as well, and developmental processes and family health. So you can see some of the areas that we tend to fund more of in FY20, and then down near the end, especially in sleep and sexual behaviors, less than some of the other areas, but a good sense of overall the type of funding that NIH has done over the current fiscal year. Next slide. So I want to shift now to the funding that we do in the Office of Behavioral and Social Science Research in terms of our co-funding. As you know, we're not a grant funding entity, so we don't directly fund, but we work with the institutes and centers to help them fund meritorious behavioral and social science research, especially when it's sort of on the cusp of their funding in terms of their ability to be able to fund it. So next slide. So if you look at those same breakdowns of those 13 content areas by the type of research that our office has co-funded in FY20, you see a pretty similar sort of breakdown, and then also a prioritization that's very similar to overall. We do a little bit more in social process disease and determinants and in healthcare and disease management areas in terms of co-funding than in some of the others. But as you can see, again, pretty much across the board and fairly consistent with the overall funding of behavioral and social science research at the NIH in general. So next slide. And if you look at this over the course of years, the green are the average from FY17 through 19, and the blue are for FY20. And just an increase overall, still pretty much the same kind of prioritization of the types of things that tend to get funded by OBSSR in terms of co-funding with the ICs. The one place you might see a little slippage this last year has been in food intake and physical activity research. But again, from year to year, these sort of modify and change as we go through different funding cycles. And sometimes one really large project that we fund fairly heavily will kind of sway these data as you move forward. Next slide. This is probably one of the more important messages about our co-funding this year. We have consistently had an increase in our co-funding to the ICs over the course of the past few years. But this year with COVID, we particularly tightened our belt for other things and made sure that we put as much money as possible into co-funding, particularly around COVID related efforts. So what you see here is an increase that's pretty substantial in the co-funding of OBSSR this year. And I expect that next year I'll probably say something like there's been a little bit of a drop, but that might be in all likelihood because we really did put a considerable effort into focusing as many dollars as we possibly could into addressing research around the pandemic. So next slide. This is just, again, sort of the number of grants that we co-funded by ICs. And as you can see, once again, there's a fair amount of consistency between this and the overall funding of behavioral and social science research across the NIH. Some of the smaller institutes tend to get a little bit more funding from us, and then again, that changes from year to year based on our co-funding efforts. Next slide. We've also been pretty active in terms of funding announcements, so let me just highlight a few of those. I won't spend a lot of time here, but next slide. This is the OBSSR-led active funding announcements for FY20. I'll talk more about the administrative supplements for the coronavirus research. We also worked with the HEAL initiative on administrative supplements to support strategies to reduce stigma in pain management and opioid use disorders. We really like, this is a supplement, the third one down is a supplement to existing T32 and TL1 training grants to better integrate behavioral and social science research with other health-related sciences. So these could be T32s in which they're mostly biomedical in nature, and the supplement would allow them to expand their curricula into more behavioral and social science aspects of the training that they do, or vice versa, so that they're behavioral and social science oriented T32s in which there's an expansion of the curricula into more traditionally biomedical areas. I'll spend a little bit more time talking about firearm injury and mortality prevention in a minute. And then we also had OBSSR-led initiatives that actually come from some of our working groups. The Trans-NIH Measurement Group put together this PAR that's out on the street on research to improve the interpretation of patient-reported outcomes at the individual patient level that can be used in clinical practice. And then our Basic and Beyond Working Group put out an initiative on development preliminary testing of health-related behavioral interventions this year. So a fair amount of work in this area. So next slide. And then if I did a slide of every behavioral and social science FOA across the NIH, I would be here well past the time that you would like to have me speak to you. So I'll keep this short. And so I just did some samples, one from each IC, and particularly ones that are trans-NIH in nature, whether they're from the BRAIN Initiative or Blueprint or HEAL or some of those other related areas, as well as just some large projects where there's been behavioral and social science research FOAs that have been developed by many of the ICs across. And again, this is just a very small sampling of the funding announcements that have been released in FY20. All right, next slide. So usually at this point in the presentation, I talk a bit about the transdisciplinary research programs in which BSSR, Behavioral and Social Science Research, has been integrated into and has worked with these programs in a significant way. And you see some of these here. The ECHO project is moving along quite well in its third year, and I think with well over 500 publications to date in the area of environmental influence on child health outcomes. OpNet released this year a social isolation initiative that I thought was particularly exciting, and we've already received a number of good applications from that as well. Our group on the Pediatric Research Consortium pulls together people on the pediatric space across a wide range of work to do the type of research that's particularly necessary for pediatric study. And again, a number of these areas in which social and behavioral scientists at the NIH, across the NIH, have been involved in these efforts. And normally what I would do is go into each of these in a little bit more detail, or at least a few. But as you all know, we've been through an interesting FY20, and the coronavirus pandemic has been sort of the crucial 800-pound gorilla in the room. So it's been the thing that we've dealt with the most. So next slide, I want to just focus on a few things that we've done at the NIH as far as behavioral and social science research related to that. So one of the first things that happened soon after the pandemic hit was a number of the institutes and centers released urgent supplemental competitive and administrative supplement requests for research that would address some of the sort of urgent COVID-19 research questions that were going on. And a number of the ICs have done that. And as you can see from the link on the bottom left is a link to our site where we list as many of those, and I think many of them are still ongoing, supplemental funding announcements. And OBSSR released its own that was a trans NIH one in nature to address a range of things, including adherence to mitigation strategies, how much risk reduction occurred from that mitigation, what the economic impacts were from those mitigation behaviors, how much the social impacts were, the downstream health impacts on mental health and substance abuse and chronic disease management and other types of things, including healthcare access, and then natural experiments to the degree possible from the variations in how various sort of mitigation strategies were rolled out by various states and counties and localities. So next slide. We also realized relatively early in this pandemic that there was going to be a rapid need for survey items that could be fielded, many by ongoing cohort studies that were already out in the field and just needed to do that. But things were moving so quickly here that we didn't have time to do the sort of data harmonization and consensus of, what are sort of best practices and best measures for measuring these various COVID-19 specific constructs. So instead, what we did rapidly was ask researchers to post their surveys as they were preparing them and getting ready to field them, and put them on two sites, on the Disaster Research Response site and on the Phoenix Toolkit site. And so in both of these, we now have, thanks to the many researchers who were very gracious with sharing as much of their work as they were doing and in real time as they were doing it. We have nearly 100 surveys that are COVID specific that are available there for others to use, in the hope that people would do data comparisons and data harmonization and borrow from other people's work so as not to recreate the wheel 800 times to ask similar sorts of questions, but be able to do better data linkages between some of these projects. Some of these surveys are in very large nationally representative samples from census and other sort of places, so RAND panel, those types of things. So there's some nice survey research here in which people can pull from survey items that they know they can then subsequently link to nationally representative samples in the future. So I just point you to that if you're not aware of it. So next slide. One of the major supplemental appropriations from Congress was $1.8 billion for diagnostic testing for COVID. And this was a large effort that resulted in 4 projects: RADx Tech, which is the rapid sort of development of new platforms and challenges for new platforms for at-home and point-of-care testing for COVID-19, RADx Rad and RADx ATP, sort of, again, kind of pushing the envelope. But the one I want to focus on is, at least during my 15 plus years at the NIH, the most substantial investment in behavioral and social science research, which is the RADx Up, or RADx in underserved populations. So next slide. So what RADx Up is about is enhancing COVID-19 testing among underserved and vulnerable populations across the US. This consortium of community-engaged research projects designed to rapidly implement testing interventions and study their implementation and uptake and follow-up, and strengthen the available data that we have on disparities, both in infection rates and disease progression and outcomes for COVID-19. The first phase of this work has already been funded. Just recently, I think just last week or the week before, was the launch meeting of the consortium itself. And the first phase of 300 million is out, but the second phase of research will be coming along in this coming year in FY21 to facilitate this. In addition to those community engaged research projects, there are also projects focused on social, ethical, and behavioral implications related to testing and vulnerable populations that I hope will generate a lot of research, not only applicable to COVID-19, but also applicable to a range of not only infectious disease research, but even more broadly, health risk behaviors and health disease processes moving forward. Next slide. One of the other early efforts that we did, which has been a little choked for money, to be frank, but we've done the best we can under limited amounts of money at this stage of the game, was an effort on social, behavioral, and economics impacts of COVID-19. And the focus here has been on effects of various mitigation strategies on reducing transmission, social and economic impact, some of the many things I talked about before already, and then interventions to ameliorate those, including more research on telehealth and digital health interventions, as well as more on community-based interventions that would ameliorate some of the health access restrictions that have been in place as a result of the pandemic and its mitigation. So there's been some really nice research there. Let me go to the next slide. Like I said, even with some limited funding, this was sort of pooled funding from existing ICs, and we had partnerships with a number of institutes and centers to pull together funding. And we also received 11 million from the Office of the Director to help jumpstart this effort. The group has been pulled together, developed a data harmonization document for all the supplements that were funded, published a commentary on this, developed and published both the digital health and community health NOCs and the PARs. So those are out there to be reviewed, and we'll be funding them sometime in FY21. But so far, I've already funded, just on the intervention side of things, 24 supplements to date. And then on the data science end of the spectrum, have funded 28 supplements to date. And we've also provided funding to the National Bureau of Economic Research to conduct research on economic impacts and excess mortality as a result of this, as well as supplements to the IHME, which many of you know because that's the data that you hear from on a regular basis in the news every day, tracking the COVID-19, both infection rates and modeling of future infection rates to look at county level disparities research on COVID-19 morbidity and mortality. So that's, I think, been a very successful project over the course of the last 6 months or so. Next slide. But we haven't just been dealing with COVID. COVID has certainly been one of the primary sort of trans NIH social and behavioral efforts that have gone on. But in FY20, we also received a specific appropriation from Congress on firearm injury and mortality prevention research. So Congress provided $12.5 million to conduct this research, to take an approach in which we looked at underlying causes from evidence-based methods for prevention of injury, including crime prevention, to do this in an open, pre-registration open code, open data way, and to make sure, of course, given the sort of Dickey Amendment, that nothing was, funds were being used to advocate or promote gun control specifically. That said, let me go to the next slide. This is the funding opportunity announcements that went out for both R61s, which are, for those not familiar, are sort of a larger version of R21s that we put out. So 400K in direct cost per year for a 2-year maximum. And we had multi-year funding authority for this. And then competitive revisions to some of the existing projects that were already out there in the field doing this research to expand them into areas where they could address firearms research moving forward. Next slide. So we managed to fund 9 projects overall with this work. As you can see, as you look at this list, it is a range of projects dealing with different types of vulnerable groups, from intimate partner violence to elderly, to people who are contemplating suicide, et cetera, et cetera. So there are a number of different groups, I think, that are involved here, as well as a number of different approaches, from more individual approaches to more policy approaches, including looking at differences in gun control policies as they impact firearm violence prevention. So it's been nice to see this nice set of projects moving forward and being funded. And our understanding at this point is in the current appropriations being considered, there may be additional funding as that moves forward as well. Next slide. But just to close on some of these various trans-NIH research initiatives in FY20, certainly some of the ones I showed you at the beginning and our COVID efforts and our firearm efforts have all been very productive trans-NIH behavioral and social science research initiatives, but there have been others. And so one of the working groups of the Behavioral and Social Science Coordinating Committee has been addressing issues around structural racism and health outcomes, especially in the aftermath of the George Floyd murder and all the things that have come since then in terms of our understanding of systemic and structural racism in the country. And so that group has already provided input to an NIH leadership group that's looking into how we address these issues, not only in terms of research efforts, but also in terms of workforce diversity, both within the NIH and more broadly extramurally. We stood up this year the Basic Behavioral and Social Science Research Council of Councils Working Group to look more specifically at what are sort of new directions and promising and emerging areas in basic behavioral and social science research. We've continued to work with our partners at the Office of Extramural Research and at Office of Science Policy on basic experimental studies involving humans, as well as with our colleagues at clinicaltrials.gov on ways that basic research can register and do results reporting in the clinicaltrials.gov platform. Our Behavioral Ontologies Development Working Group of the Behavioral and Social Science Research Coordinating Committee just commissioned this September, just before the end of the fiscal year, a National Academy's consensus study that will be going on this year to lay the groundwork and hopefully help map directions for how to develop behavioral ontologies more consistently, so we can be more consistent about what we mean by our various constructs and how they're defined, and build a more cumulative science as a result. We awarded T32s, the Training of Advanced Data and Analytics for Behavioral and Social Science Research, which I always like the acronym TADA. That has been launched and funded, integrating behavioral science and data science in these training programs. And they've already been meeting, and they also already have ongoing open presentations and training lectures that you can certainly listen in on to, and so that they can provide training not only for their trainees, but also for a broader audience as well. Back in January, we've finalized and signed off on license agreement with the National Death Index at the National Center for Health Statistics that provides NIH-supported investigators with more ready access to the National Death Index when they're doing data research using those mortality data. This essentially makes it so instead of having to pay on a per record basis for each investigator, instead of that, what people will be able to do is pull that data, and the NIH, for any NIH-supported investigator, is paying the national index to cover those costs, and also trying to make this a little bit more of a streamlined process along the way. And we've been really fortunate to have really good partners at the National Center for Health Statistics in getting this done. And then a work group led by Christine Hunter and Sylvia Choi have been working diligently on a vaccine communications best practices, pulling from the prior research that we know about how to communicate about vaccinations and encourage vaccinations and address vaccine hesitancy, and address that and moving forward, make that available to as many people as possible, so that we're communicating as best as we can about how to encourage people to get vaccinated when the vaccines eventually come out. Next slide. So to end, last year I was fortunate that I didn't have to name anybody because things had been so quiet and so stable at OBSSR. But it's been really nice to have some new people added to our group this year. Desiree Bellis comes to us from NHLBI and their Citrus program in our health policy analyst group. So it's great to have her. Caitlin Bergdorf, who you've heard throughout these 2 days moderating our question and answer session, is our new AAAS fellow with a neuroscience background from Cornell. So it's great to have Caitlin on as well. And then coming soon in January, Sarika Parasaraman is currently at HRSA and will be coming to us from there. And Kristen Brethel Horowitz is at a postdoc at the University of Pennsylvania, and she'll be joining us in January as well. So it's great to have some additions to the team. We have a great team at OBSSR and some wonderful staff that I am privileged every day to work with. And we've been able to sort of add to that group in some important ways. So it's nice to have these new people along. And then the final slide, I want to particularly thank the people who pulled this together. This is not an OBSSR effort. This is a Behavioral and Social Science Research Coordinating Committee effort for us. So that's the key thing. And the people that you heard present were nominations from across the ICs of people that they thought really sort of exemplified the sustained and profound research impacts that their grantees are doing. So it's been great to have our presenters do that today. These are the 4 people who really sort of pulled this festival together. And I particularly want to do a shout out to Dana Green Schlosser, who has led this effort and behind the scenes has pulled all this together and kept us all on track and made sure that this came off wonderfully. So to Dana and Michael and Liz and to Caitlin, thank you all very much. That's my last slide. Caitlin, I wasn't sure, are we going to do questions or do we just close up?
I think we're skipping the questions for now, and I think you can close this up.
All right. Sounds good. So I want to thank all of you for taking the time to do this today. Once again, thank our presenters who made wonderful presentations that I think exemplify the type of behavioral and social science research that the NIH supports. And I hope all of you have a quiet and restful holiday season coming forward. I know it's going to be a little different than usual. I hope it'll be a little different than usual. I think it's particularly important that we're staying safe as our infectious disease colleagues work furiously to get us close to vaccines being available in the next year coming up. So I want to thank you all again. Thank you for your time and your commitment here. It's been much appreciated, and we'll close this now. Thanks very much.